Okay, so are you all back? Why are the cameras are off? Marej, Hania, Khanza, why your cameras are off? Okay, so now we are going to see again anterior posterior pattern formation or axis formation. And we are going to continue after maternal genes. So you remember we talked about the famous segmentation screen for body patterning by Eric Weishaupt and Kristen Noslin-Follard, in which they categorize their mutants into different classes. So one of the classes of the mutants was named as gap genes. Now, the expression of these gap genes, it is discovered or that, they are, they coincide with specific broad regions within the anterior posterior axis. And their expression is still coinciding when embryos are yet to be cellularized. And these are actually one of the first zygotic genes which become active. So maternal genes, they activate these genes. These are the first zygotic genes to be transcribed. And all the gap genes, they are also transcription factors. So what we learned in case of maternal genes, that bicoid gradient is formed in anterior posterior axis formation like this. If this is bicoid, we said there will be also nanos caudal on the posterior side. If you have mutations in gap genes, there are big regions in the embryo which are absent. And that's why they're also called gap genes, because absence of gene products of these proteins, they leave big gaps in embryonic development. And these are also short-lived. They are transient in nature. As I said, they are transcription factor. Once they perform their function, they disappear. Some of the examples are given here. For example, giant is a gap gene which is expressed in very discrete region. So in contrast to the maternal effect genes, they don't have gradients. Krupal has very discrete region. NEBs have discrete region, and then tail. So all the gap genes, if you pay attention, they are in discrete regions. Now, a gene called hunchback, we already talked about hunchback. It is maternal effect gene as well as early gap gene as well. Okay, so what we learned that bicoid is actually the one which activates hunchback. Hunchback comes in as maternal effect messenger RNA as well, and then wherever we see bicoid, we also see hunchback. But bicoid as transcription factor also binds to hunchback gene, zygotic gene, and activates its transcription as well. So zygotic hunchback is actually one half of the embryo, okay, because it coincides with concentration of the bicoid. Okay. And you can superimpose the concentration of hunchback maternal effect protein as well as the zygotic hunchback, because zygotic hunchback will respond to the concentration of bicoid. And hunchback from posterior side, we also learned, is suppressed by nanos in the posterior end. If you increase the concentration of bicoid to 6 doses, sixfold, you see a proportionate increase in the hunchback protein concentration as well, which means hunchback protein is actually activating zygotic hunchback in a proportionate manner. Now the question is, what is the relationship between hunchback and other gap genes? Or one can also ask question how these expressions of gap genes in very discrete regions is established, knowing we have these gradients, knowing we have bicoid gradient, we have hunchback gradient, we have gradient from posterior effect genes as well. So how then we, instead of seeing some gradients, we see very discrete expression pattern in discrete regions. So regarding regarding Krupal is a transcription factor in gap genes. What we have learned that it responds to a very, very specific concentration gradient or threshold of hunchback. So if you move this threshold of hunchback either forward or backward, you will move Krupal back forward or backward as well. For example, the proof is if we reduce the concentration of bicoid in the bicoid mutant, very little bicoid is present. Okay. Now, when there is little bicoid present, it means hunchback will also be reduced, because zygotic hunchback is active due to bicoid. Now, due to absence of bicoid, the concentration of hunchback goes down, and this threshold, which was this one, has moved here. So previously in wild type, beyond this region, the hunchback was high. Now that is diminished. So this threshold gradient and this one, you can see now Krupal going this way. Is it clear so far? Yes or no?
Yes, sir.
So there is a clear dose dependence of Krupal gene expression on bicoid and hunchback concentrations. Now, what we have also, we are going to see that the anterior border of Krupal is 4 to 5 nuclei, posterior to the nuclei that express giant. So this is giant, and this is Krupal. So here there's just 3 to 5 or 3 to 4 nuclear border there. So it is that discrete, you know, it's not just some random effect. Uh, Development is a very serious business. Development is very precisely engineered business. When it says 4 to 5 nuclei, it is 4 to 5 nuclei. And this 4 to 5 nuclei and this, what we call the borders or whatever, they are relying on these tight concentration gradients, thresholds. If you increase slightly beyond this threshold, you are moving this, maybe 2 nuclei on this side, and the whole developmental plan will be messed up. Now, the whole anterior posterior axis formation, what we have learned in last 4 or 5 decades is that, you know, there are actually unique regions, and these unique regions are actually based on the overlapping uh, gradient concentrations uh, of different transcription factors. You know, and soon we are going to see how these overlapping concentration gradients of different transcription factors, they impact heart development or the fly development. But before we move there, let's talk about how do we know that the hunchback promoter responds to Becoid or any transcription factor. And there is an experiment which shows that, so this is the normal promoter, hunchback promoter. You fuse it with bacterial lag Z gene, or you can think of GFP, okay? And you make transgenic embryos. And it shows the staining for lag Z gives you normal expression pattern of hunchback. Now, if you start truncating the promoter, so this was the wild type promoter. So let's say this is the full length wild type promoter. You make different truncations like this promoter, then only this promoter region, then an overlapping region, this one, then maybe a region where we have this one, then there's deletion, then it continues this thing. So you can make as many truncations. And you fuse with each one of them, you fuse leg Z and make transgenic flies and stain for expression pattern. Here you can see uh, expression pattern is disturbed. Here, everything is gone. Okay, so there's an indication that there are specific binding sites, there are specific regions within this promoter which are crucial. For example, maybe this region was very important. When you have this one only, everything is gone. All this was important. So this, we call this promoter deletion experiments, and we, after deleting different regions of promoter, we fuse with different reporter genes. Now, after gap genes comes segmentation genes. But before we go there, let's first learn about what is a segment in fruit flies. Okay, this is very important. Before we go to, after gap genes comes the pair rule genes. And they are expressed in very specific parasegments in the fruit fly early embryonic regions. But before we go there, we need to learn about relationship between segment or parasegment, how you define as parasegment, and what is the relationship between parasegment and segments. So as cells become cellularized or embryo becomes cellularized, um, then they start, these individual cells, they start grouping in different regions, in the head region, in the thorax region, future thorax region, and the future abdominal region. But long before that, the earliest sign of segmentation is just these 14 stripes. This is still within first few hours of development, and the earliest sign of segmentation is these, if you count, these are 14 stripes. And this is the first sign of segmentation. Slightly, so this is the early stage embryo. At late embryo, these parasegments are called segments. And here you can see C1, C2, C3. These are the head regions, T1, T2, T3, thorax region, thorax 1, thorax 2, thorax 3. Abdominal segments, A1, A2, all the way to A8, so total 14. Now, you have to understand the relationship between these early 14 parasegments and adult segments. And these, when I say adult segments, they are still within the embryonic stage, the first 12 hours or so. So these 14 parasegments, they appear as transient grooves on the surface of embryo after gastrulation, these 14 stripes. And these grooves are referred to as PS1, PS2, PS3, parasegment 1, parasegment 2, parasegment 3, all the way up to 14 parasegments. And parasegment is actually fundamental unit of segmentation in Drosophila. Now, what is a segment then? What is this one? So segment is actually posterior region of a parasegment and anterior of the next. If these are the 14 parasegments, 1, 2, 3, 4, 5, 6, 7, all the way to 14. So posterior region of parasegment 1 and anterior region of parasegment 2, they are going to make C2, segment C2. And since there's no anterior of, there's no other, so C1 is just consisting of posterior region, anterior region of parasegment 1. Now, what is important that you don't confuse posterior of parasegment 1 and anterior of parasegment 2 when they are an adult segment, when they have made C2. C2 will have its own anterior and posterior. Now, previous anterior of parasegment 2 has become posterior of compartment, posterior of segment C2. Because this is a segment, cells in this segment, they have compartmental boundary not only to the next one, but within. These cells, anterior, which were posterior of parasegment 1, and posterior of C2, which were anterior of parasegment 2, they are never going to mix with each other. We call them compartmental boundaries. These cells, they have committed to a specific cell lineage. Within that lineage, the anterior cells and posterior cells, there is a hidden boundary. We call it compartmental boundary. It's not a brick wall. It's not a membranous wall. Cells are lying next to each other. And while I'm telling you, while I'm trying to explain you, मेरे दिमाग में कुरान की एक आयत आ रही है जिसमें वो कहता है कि वो दो दरिया हैं जो साथ साथ बहते हैं, दो समंदर हैं जो साथ साथ बहते हैं, दोनों की अपनी अपनी आइडेंटिटी है। दोनों में कोई बैरियर नहीं है, लेकिन वो दोनों मिक्स नहीं होते। Many people have shown this. वो कोई जाता है कि जी फ्ला ओशन है, फ्ला ओशन है, कि जी ये वाली आयत का ये मतलब है। कुरान का परत परत खोलते जाएं तो एक आयत के आपको लाखों हजारों जो है ना वो मीनिंग मिलेंगे। So while I'm teaching you this in last 11 years, it never came to me, but this is for the first time it's coming to my mind that हो सकता है वो इसकी तरफ भी इशारा कर रहा, उसका उस आयत का एक मतलब ये भी हो। Because these cells throughout the development of fly, they retain their individual identity. They grow, they divide, but they never ingress in each other. They never are going to mix up with each other. And we will learn about the consequences of this mixing if they mix by mistake. Anyway, so do you understand the relationship between segment and parasegment? Now, one of the early selector genes, very early selector gene. Selector genes are the ones in developmental gene. There's also selector gene hypothesis. We name those genes as selector genes, which give specific identity to a cell type. And one of the earliest selector gene is engrailed. Engrailed is expressed in all the anterior regions of each parasegment. If there are 14 parasegments, anterior region of each parasegment will be expressing engrailed. So where engrailed will be in the adult segment then? क्यों भाई?
Sir, can you repeat your question?
Where you will find engrailed in an adult segment of fruit fly?
In the posterior region.
Very good. Because the anterior of a parasegment has contributed to posterior of a segment. If I ask you a question, I give you fruit fly imaginal disc, okay? Let's say I give you wing imaginal disc, I give you halter disc, I give you leg disc, and I ask you, use whatever tools and show me under the microscope, because this imaginal disc looks same. I ask you under the microscope, which region is posterior and which region is anterior? What will you do?
So we can stain it with anti-engrailed and see if it's being expressed at the posterior. So that will be उससे पता चल जाएगा कि anterior कौन सा है, posterior कौन सा.
Very good. So you will simply do an immunostaining experiment. I told you, you have to learn development this way. Otherwise, you are learning facts, and facts are not useful. So you will simply do immunostaining. You will use antibody against engrailed stain imaginal disk. And what we will see, we'll see staining like this, maybe. Wherever we will see engrailed staining, we will say, hey, this is the posterior region of imaginal disk. Okay. Now, let's talk about now pair rule genes. So what we have talked about, we have talked about maternal effect genes, activating gap genes, and now the pair rule genes. Pair rule genes, they actually express in alternate stripes. And there are two pair rule genes, two famous pair rule genes. One is called even skipped, and the other is called Fushi-Tarazu. Even skipped, it defines the odd numbers. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Even skipped is going to expressed in odd stripes, 1, 3, 5, 7, and so on. All the odd stripes will show you staining of even skipped. Now, even skipped, as the word suggests, if you have mutant in even skipped, once you will have all the odd stripes gone due to mutation of even skipped, you will be left with just 7 stripes. So even numbers are gone. The Fushi-Tarazu, as compared to even skipped, it defines the even numbered parasegments. So each of the pair rule gene is expressed in alternate stripes, 7 stripes along the embryo. This is staining pattern of even skipped and Fushi-Tarazu. So here, this is, for example, your 1, 3, 5, 7, 9, 11, 13. These are your even skipped stripes. And these ones are 2, 2, 4, 6, 8, 10, 12, 14. These are Fushi-Tarazu stripes. They are, and look at the beauty. So discrete expression pattern. They are not mixing with each other at all. And these are few nuclei. And remember, like those two oceans, cells of these compartments, they are never going to mix with each other throughout the life of the organism. They will, from here, look, from here, they go all the way to adulthood. They contribute to such complex organs and tissues, but they never mix with each other. So let's understand the gap gene activity, because it's getting complicated now. We said we'll look at expression of gap genes and the maternal effect genes and pair rule genes in a context of development. How do we see? I mean, we were wondering about how discrete stripes of gene expression are formed. We said, okay, there is concentration gradient. Now, how expression pattern is restricted to just this 3-4 nuclei stripe? This graph tells us, just tip of the iceberg. This tells us how this phenomenon might be possible. This is about the second stripe of even skipped. Second stripe of even skipped is in parasegment 3, which is here. And what we have learned over decades is that, it responds to all these actions of its predecessors, the genes, maternal effect genes and gap genes. For example, here we have the hunchback concentration. We have the Bicard concentration. We have the giant, and we have the Krupal. Where you will have least concentration of the gap genes, and this threshold of the hunchback, we will have even skipped peak. But here, the first stripe, even skipped, will be different because here you have high concentration of giant, hunchback, and different concentration of Bitcoin. And if you move to fifth, a different concentration of Krupal, so a different mechanism will be there. So, but for the stripe 2 of the even skip, this is what we know, that it requires least concentration of the gap genes and a specific threshold for hunchback, etc. And this is the promoter region of the even skipped. So even skipped promoter region. You see there is Bicard binding site. So this is Bicard DNA binding site, this one, this one, this one. Then hunchback. And then you have giant and Krupal binding sites. So the coordinated action, a combinatorial action of these transcription factors, which involve maternal effect, which involve gap genes, make it possible. And if you have paid attention to the coordinates of the promoter, it lies between minus 1070 to minus 1550 base pairs. These ones, the Bicoid and hunchback, they play their role as activators, and these ones play their role as repressors in this particular context. Now, before I move on, I would like to ask question. Shajil, one by one answer these questions. Shajil, what do you understand by what I just pointed out? Minus 1072 minus 1550 base pairs. What do you understand there? Shajil. G Nurulan.
Sir, I'm not sure I understand.
G कौन है? Kanza. Marriage.
Sir, it means that the DNA sequence that these regulatory proteins actually bind to lies between these certain positions.
G Savera.
I'm not sure.
G Amina.
I think, sir, these positions are relative to the promoter, either upstream or downstream. I'm not sure.
What do you mean by upstream or downstream? I don't know. What do you mean by this?
1070 base pairs upstream or downstream of the promoter.
Very sad state. Momina?
So I think if this promoter region lies between minus 1550 and 1070, and this is like before the gene.
You all are juniors. In 1 year, you are going to finish your degree and you don't know, very basic. If I write down here +1, what it means?
The protein, the gene which
Diha,
are transmitted.
Yes, sir.
what is
So it's
+1?
so that's basically where the starts. That's the start site where
Start site.
where the transcription will start.
Yeah, this is the transcription start site. Now, where will be minus 170? 1070.
That will be the upstream of the site within the promoter.
So, bacho, this is transcription start site. +1 means from where RNA polymerase start making messenger RNA. Anything, Amina, anything above this, you are saying, I don't know, you're upstream and you're downstream. Anything below this is downstream. And anything above this is the upstream. So this will be -1, -2, -3, -4, -5. You keep going. And we're talking about -1070 to -1550. This is usually a eukaryotic promoter is within first 100 base pairs, -100 up to -100. We have these -35 sequences and -10. Okay. This is even upstream of the promoter regions. Some regulatory sequences. Is it clear to all?
Sir, can I ask one thing?
Ji.
So basically, जो आपने अभी बात की है, so it means that the upstream, there are multiple sites where these regulators can bind, but in different paths, like it's not binding on like all of these regulators. They're not binding at the same time to each of these sites. Is that what you mean? Because there are more sites available, and when there's a gradient and there's a higher concentration of a certain transcription factor, it's going to take or bind to the maximum sites. This is what I'm getting. Can you please explain this a little bit?
So if you go there, if we go there, and now even skip promoter, which we just saw, it's present in every cell. Even skip promoter will be same gene, same promoter is present in cells which are here, which are here, which are here, which are here. Okay. Now, these coordinated action of all these different players are going to determine whether even skipped will be active here and silent here. Here silent, here active. Here silent, here active. Promoter is same, but it will be the coordinated action. Bacoid, hunchback, and Krupal giant. Or if we move maybe to the earlier positions, we may have, or to the, we may come across nerves as well. It may also contribute. Okay. So we don't know precisely how many proteins molecules of bacoid should be there, and then whether it will be, what kind of effect it will have, whether it will overrule these two repressive effect. It looks like when we have least concentrations of giant and Krupal in this cells in this region, they will be not effective. They will affect. They will bring the, but we cannot say this for every stripe of even skipped. Clear?
Yes, sir.
Any other question? Any other question?
Sir, I have another question. In the
जी,
slide that you showed, आपने discuss किया था hunchback promoter experiment when we were using the promoter fused with the LAGZ gene and then we were checking for the expression after staining it with. So उसके अंदर I had a question कि अगर आप उस slide पे जाएं,
this
yes.
one?
So yes, exactly. So basically, in the normal promoter, we are saying that the expression is restricted to the anterior side, but when there's a partial promoter, we see that there's an expression on the dorsal and ventral side as well. And when the promoter is deleted, there's this expression on the that's the ventral side. So can you explain like why when we are like affecting the promoter, the expression is going like it's happening this way? Because this is what like this is showing that the promoter is kind of active in these parts. This is what I'm getting from this. So can you explain like why this is happening? Is it because of what we just like went through, जहाँ पे आपने बताया कि it's a coordinated expression? So this is an example of that.
Yeah, because by deleting the promoter region, you are eliminating some of the binding sites of activators.
Okay.
Clear?
Yes, sir. I just wanted to clarify this one.
Any other question?
So I just wanted to ask that you said that जो DPP है, it is being expressed in the first few hours throughout the embryo, and it is then limited to one part of the embryo. So is it known what it is doing in the first few hours throughout the embryo? Because
No, I did
like
not say DPP is expressed only in first few hours of embryo. I did not say this. DPP,
throughout the embryo, in the
yeah,
first few hours.
yeah. So DPP protein is present throughout in the dorsal region. Okay, and it's a maternal effect gene. To be more precise, in the early embryonic development. Later on, zygotic transcription also takes place. So in the first few hours, when the dorsal ventral axis is formed, DPP is actually counteracting the spread of dorsal in the dorsal region. Because as soon as Spätzle gets cleaved and toll signaling is activated, you know, toll is also uniformly distributed all across the cell surface membrane. And if you have Spätzle, you know, you may lead to pushing dorsal by mistake in all the nuclei. So DPP restricts dorsal only up to neuroactiva. Any other question?
Sir, I have a question.
जी
तो जब भी आपने आखिरी स्लाइड में दिखाया था प्रमोटर के साथ बाइंडिंग साइट्स में, तो व्हाट इफ लाइक जो हमने ग्राफ देखा है उसमें एंड में तो खाली क्रुपल रह जाता है, तो इज दैट इनफ टू एक्टिवेट फॉर द लेटर सेगमेंट्स, टू एक्टिवेट द इवन साइड?
Which one you are talking about? एंड में क्रूपल कहाँ पे रह जाता है? This one?
ये जो कंसंट्रेशन वाला ग्राफ है ऊपर वाले में.
This one.
Yes, over here in the end we only have Krupal. तो जो आप promoter वाली स्लाइड है नीचे वाली, उसमें like there are 4 binding sites, right? जो आपने बताया कि बिकॉइड hunchback एक्टिवेट करता है, giant और Krupal repress करता है. जो later segments हैं, जो segment 13 है और 11 है, वहाँ पर किस तरह से होगा? Because ये वाली बात तो present नहीं है.
Krupal is only in very discrete region here. ठीक है? Krupal is only present in this region. So hold on. Krupal is only present in such a stripe. If you go downstream, you will have different genes in the embryo, different gap genes, which will be regulating even skipped. It's never only giant and Krupal.
Okay.
Any other question? So if there are no more questions, thank you very much. I wish you a good day there. And also, I would like to apologize for skipping last 2 lectures because I had a high grade fever. I don't know, all of a sudden. Luckily, I got cured within 4 days or so. So it wasn't the usual what we know as COVID, etc. But just I took the precautions so that I restricted my movement. But Alhamdulillah, I was fine within 3-4 days out of this danger zone. Oh yeah, one more thing I wanted to announce. In the evening today, we have a seminar. Again, don't miss this seminar. This is about transcriptomics. This is about how you use global gene expression or transcriptome of an individual and how it can be used as a personalized medicine. This is a very exciting area. The professor is from Georgia Tech. I think you must have seen the flyer or the email notification. So I expect each one to be there. I think you must have enjoyed the Mitzi Kuroda seminar last time I saw, and I was very happy. Many of you were there, except one or so. I was looking at the attendance sheet. Don't miss these seminars. Being juniors, these are mandatory for you, and they are part of your Dev Bio. They are part of your 415 course. Okay. What we are covering in tools or methodology in the recitation, these seminars will help you in understanding and applying those tools because these big scientists, they are using those tools to understand development as well. So I look forward to see you. I think it's 6 in the evening, I believe, today. Yeah, it's 6 in the evening. I look forward to see you there. Bye-bye.